Hello, my dear students. Welcome you all to Sri Jaindra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last video, we started with physiology of muscle contraction. So today, we will be completing the topic. There are some known and unknown facts which are remain to discuss. In the previous video, we discussed about the sliding filament theory, the biomolecules, especially the proteins which are required for muscular contraction, that is contractile proteins. Today we are going to discuss some important and related terms, especially made for certain competitive examinations. So starting with muscle twitch and tetanus. These two different terms, muscle twitch and tetanus. So before talking about muscle twitch or tetanus, I would like to tell about refractory period. What is refractory period? It is the time period during which a muscle fails to respond for a second stimuli. Very important, for a second stimuli. Means if the muscle is already carrying a stimuli, okay, and that stimuli is the stimuli is under the passage, the stimuli is not crossed the synapse of one muscle, then that muscle cannot take the other stimuli. Okay, so this is the refractory period, or we can say refractory period is a time interval between two consecutive muscular contractions or two consecutive muscular excitatory phases. Okay, so there is a time period during which a muscle fails to respond for a second stimuli. Now, a very important fact, in any muscle, if the refractory period is short, it means it can take or it will try to take multiple stimuli at a time. Okay, so skeletal muscles, they have short refractory period. I should say the shortest refractory period. And because of having shortest refractory period, they didn't get time to relax. Okay, and hence they undergo, hence they can lose all the calcium content and can enter the condition of tetanus. Okay, is it understood? Tetanus, it is sustained contraction due to repeated stimuli. Sustained contraction. So tetanus is basically a sustained contraction due to repeated stimuli. The muscle is contracted for a prolonged period without any rest or relaxation. And very importantly, a continuous stimuli is required for the maintenance of tetanus. Okay, that is why it will undergo sustained contraction. While the muscle twitch it is when I'm talking about a single muscle twitch, so it is a single contraction followed by relaxation. Due to stimuli. Okay. Now, muscle fiber contracts only once if it is stimulated by a single nerve impulse or by a single electric shock of threshold stimuli. Okay. This single isolated contraction of the muscle fiber is called single muscle twitch. Okay. After a twitch, a muscle fiber relaxes. It relaxes. So, as I have already told you about the definition of threshold stimuli, I just repeat my words. For a contraction of a muscle fiber, there is always requirement of a minimum and specific strength or intensity of the stimulus. So it can be said as a minimum, specific, strength stimuli. It should be minimum, it should be specific. Okay. So the specific strength of a stimuli, minimum and specific strength of a stimuli required for contraction. Required for contraction. It can also be understood as the minimum intensity of the stimuli or a nerve impulse for excitability. For excitability. Okay. The threshold stimuli. Okay. So tetanus, muscle twitch, threshold stimuli. Understood? Now, the next term what we are going to discuss is all or none law. If a threshold stimuli is applied to a muscle fiber, it can contract maximally. This law is not applicable for complete nerve. This law is not applicable for sorry, not complete nerve, for complete muscle. Okay. Why I am telling this? Because the fact is, in a single muscle, we are having different muscle fibers. These muscle fibers have different threshold stimuli. Okay, so all or none law is basically meant for a particular muscle or a particular whole neuron. Okay, it is for a muscular cell. Okay, so if a threshold stimuli is applied to a muscle fiber, it can contract maximally. That is very clearly understood, no doubt about it. And this law is not applicable for complete muscle because all the muscle fibers, they don't have same length, they don't have same intensity to carry on with the same threshold stimuli, right? So these are some points which you have to remember. Okay. Now, myosin versus actin filaments. What is the difference between myosin? What is the difference between actin filaments? Okay. Myosin filaments, they are only found in A band of sarcomere, right? They are only found in A band of sarcomere. They are thicker but longer, thicker and longer than actin filaments. Cross bridges are present. Cross bridges are present, and hence they have rough surface. They have rough surface. Okay. The number of myosin filaments is lesser than actin filaments. Okay. So we can say the three myosin filaments, they surround the actin filaments. Three myosin filaments surround actin filament. Okay. They are free at both the ends. Okay. And they consist of two proteins. One is myosin and the other one is meromyosin. Okay. They do not slide during muscular contraction. Do not slide during muscular contraction. Then is actin filaments, which are thin myofilaments. Myosin filaments are thick myofilaments. So the myosin filaments are thick myofilaments. They consist of, I should say, they consist of. They are only found in A band of sarcomere. They are thicker and they are longer than actin filaments. The cross bridges are present and hence they have rough surface. They are lesser than actin filaments. Only three myosin filaments around one actin filament. They are free at both the ends. They consist of two proteins, myosin and meromyosin. They do not slide during muscular contraction. Okay. Now comes the actin filaments, which are thin myofilaments. Okay. Actin filaments or thin myofilaments, they are present both in A band and I band. They are found in both A band and I band. Understood? They are found both in A band and I band. They are thinner and shorter. Shorter. Okay. Cross bridges are absent and hence they have smooth surface. And hence they have smooth surface, right? Then they are more in number in comparison to myosin filaments. Six actin filaments surround one myosin filament. Understood? Next, they are free at one end and are joined to Z line by other end. Yeah. So these actin filaments, they are free at one end and are joined to Z line by other end. Okay. They consist of three proteins: actin, tropomyosin, and troponin. And troponin. Okay. And they slide into H zone during contraction. Okay. Is it understood? So this is the actin filaments, which are thin myofilaments. Okay. They slide into H zone during the muscle contraction. Okay. Rest all the proteins, everything is discussed. Okay. About detailed structure of myosin filaments. Okay. So this is the myosin versus actin. That what are the differences, or what are the basic facts you should know about myosin and actin filaments. Okay. Now types of muscular contraction. When we come to types of muscular contraction, so there are basic two types of muscular contraction. One is the isometric contraction. Okay, isometric contraction. And the second is isotonic contraction. Understood? Isometric contraction, iso means same and metric means length. Okay, mean there is no change in length. Oh, seriously? If you are contracting something, so how it can be possible that length will not be changed? Okay, so that is why it's a very important term to understand. Isometric contraction, no change in muscular length during contraction. Okay, because the work done is zero. Work done is zero. Okay, mean the muscles is generating a force, but there is no change in length. Muscles they are generating a force, but there is no change in length, no displacement. No displacement means no work is done. Okay, so that is isometric contraction. Okay, force is generated, but there is no displacement. Understood? So this is the isometric contraction in which work is zero. Okay, work is zero. The force is generated, but the fact is there is no change in the length, there is no change in, or there is no displacement actually occurring. Okay, so this type of contraction is known as what? Isometric contraction. Okay, during this contraction, the tension is increased. The tension means we can say the tension on muscle is increased. Okay, the tension is increased. Example, if you pull any heavy object, okay, this is isometric contraction. Isotonic contraction. What isotonic contraction says? Mean iso means same and tonic means tension. So here the length will change but the tension remains same. Okay. Tension is basically the force generated by muscle. Okay. For example, this is connected. Okay. So the tension is working opposite to the weight. Okay. Tension is the force generated by muscle. Example, lifting of an object. So here tension is generated. Okay. But that tension remains same. Length changes. length of the muscle fiber changes. The muscle fiber gets shortened up during contraction. Okay. For example, bending, simple bending of an arm, lifting any object, lifting any object. So these are the types of contraction: isometric contraction and isotonic contraction. Okay. Now, during sternus exercise, the deposition of lactic acid may cause fatigue. That you know, I will be telling more. Okay. There is an important term I should mm, tell you: trip. Trip or trepe is a repeated contraction due to subtetanizing stimuli. Okay, so trepe is basically a repeated contraction which is because of subtetanizing stimuli. I mean, a stimuli which is, you know, above the threshold, but it is not equivalent to the potential of potential for causing tetanus. Okay, so the stimuli, the potency of that stimuli is subretinizing. Okay, so it will create multiple contractions at a time. Okay, but it is means slightly less than the tetanus. Okay, slightly less than the tetanus. Okay, so when we plot a graph, okay, then the muscular twitch graph goes like this. Muscular twitch. Okay, the tetanus graph goes like this, and the trepe graph goes like this. Very random. Okay, so this is trepe, this is tetanus, and this is muscle twitch. What this graph is actually showing that the muscular contraction will reach to its peak and then it will retard, it will relax. Here the muscle contraction will be at peak, but the relaxation get delayed. Okay, sustained contraction, sustained contraction. Okay, sustained contraction. And trapeze sustained contraction followed by frequent relaxation. Frequent relaxation, you can see. This is trapeze. Okay. Now moving to next, skeletal muscle fibers. Now we are going to talk about the skeletal muscle fibers. Okay. The skeletal muscle fibers, they are basically of two types. We have to study about two types of skeletal muscle fibers. Red fibers and white fibers. Red fibers, they are the type 1 muscular fibers, skeletal muscle fibers, and they are type 2. Okay. First point. They are narrow in diameter, narrow diameter, broader diameter. High content of myoglobin, no myoglobin. White fibers, they don't have any myoglobin content, right? The number of mitochondria in red fibers, type 1 fibers, is high. Low number of mitochondria. Chondria. Okay? Then the red fibers, the type 1 fibers, they have less smooth endoplasmic reticulum. Smooth endoplasmic reticulum. More smooth endoplasmic reticulum. They are slow and oxidative fibers. They are fast, okay, and glycolytic fibers. So red fibers, they are slow and they are oxidative and they undergo the content of them undergo complete oxidation. But the white fibers, the type 2 fibers, they are fast in working, but they are glycolytic, mean they can only undergo the glycolysis, that is, cytosolic breakdown of food nutrients, food materials. Okay. So that is why as the cytosolic breakdown of glucose, okay, so they are basically undergoing through the journey of anaerobic change and aerobic oxidation, and that is why they will lead to more amount of lactic acid accumulation. Okay, so the white fibers, they will lead to more lactic acid accumulation. So lactic acid does not accumulate quickly. Quick accumulation of lactic acid. Understood? They do not fatigue easily. Fatigue is They get easily fatigued. Okay. Examples are muscles of back, flight muscles of long distances, flying birds. Okay. Muscles of eye, muscles of eyelids, flight muscles of short distance flying birds such as sparrow muscles. Okay, red muscle fibers are more developed in marathon runners. Okay, white fibers, I mean, if a person is going for a marathon running of around 42 kilometers, okay, so they could have developed more red fibers. But the one who is going for a 100-meter sphincter race, so red fiber or white fibers will be developed. Okay, understood? Glycolysis is the main energy source for white fibers. Glycolysis is the main energy source for white fibers. Okay, so these are the differences between the red fibers and the white fibers. Okay, now my dear students, I will be going to discuss about the requirement of different biomolecules. Okay, in muscular contraction. What is the role of different biomolecules in muscular contraction? There are a number of biomolecules which are involved in muscular contraction. First is muscle proteins. Muscle proteins we have already discussed. Myosin, actin, tropomyosin. Troponin. They all play a significant role in the muscular contraction. Okay. Then myoglobin stores oxygen. Okay, myoglobin stores oxygen. Smooth endoplasmic reticulum stores glycogen. So glycogen is a very important nutrient, okay, which is required for muscle contraction. Okay, lipids. Lipids are the neutral fats. Okay, then high energy phosphates such as ATP and phosphocreatine. Okay, ATP and phosphocreatine, which provides energy. Now, phosphocreatine, the presence of creatine phosphate or phosphocreatine is characteristic feature of vertebrates. Okay, presence of phosphocreatine is a characteristic feature of vertebrate. Of vertebrates. It is used as an energy source. Inorganic substances like potassium, which is at the highest amount, potassium. It is the principal mineral of the muscle, highest in amount. Okay. Sodium, calcium, magnesium are also there. Chloride ions, phosphate ions. Okay. Enzymes, myosin ATPase. These are all the different substances which are required during muscular contraction. Okay. These are the role of biomolecules in muscular contraction, muscle proteins, myoglobin. The smooth endoplasmic reticulum stores glycogen and lipids, high energy phosphates like phosphocreatine, which is a characteristic feature of vertebrates. Then potassium, which is a principal mineral and highest, in amount. Na plus, calcium, Mg2 plus, Cl minus, PO4 3 minus. Okay, when we talk about enzymes, so myosin ATPase is the principal enzyme. Energy source for muscular contraction. Adenosine triphosphate ADP, second creatine phosphate, okay. Creatine phosphate basically it is stored in muscles, synthesized in liver, which releases phosphate ions. Okay. During any activity, creatine phosphate CP combines with adenosine diphosphate in the presence of creatine kinase to form creatine plus ATP. So this is the reaction during any activity. The creatine phosphate combines with ATP adenosine diphosphate in the presence of creatine kinase to form creatine and adenosine triphosphate. Understood? The creatine is converted into creatine phosphate during rest. Phosphate. But there is some loss of creatine and its anhydride called creatinine is formed. Okay, but loss of some creatine gets converted into its anhydride called creatinine. Okay, and this creatinine it passes out in urine. Okay, creatine is formed in liver from three amino acids. It is formed in liver with the help of three amino acids. With the help of three amino acids, glycine, these are glycine, arginine, and methionine. So creatine is basically formed in liver with the help of three amino acids, glycine, arginine, and methionine. Okay. Creatine phosphate is basically formed at the condition of rest when creatine is converted to creatine phosphate, but loss of creatine gets converted into its anhydride creatinine. Creatinine is passed out in urine. Okay, that is why the persons which are taking high creatine diet, the bodybuilders, they are recommended to take high water intake or large water intake to clear out the creatinine. To clear out the creatinine. Okay, it is formed in liver with the help of three amino acids: glycine, arginine, and methionine. Okay, Cory's cycle. Now we are going to discuss Cory's cycle. It was given by Cory's cycle was given by Carl Cory and Georgi Cory. In the year 1947, both of them really received Nobel Prize. Okay, so Cody cycle is basically a link which connects that how the lactic acid can be resumed to, resumed to form energy. How the lactic acid can be utilized as a energy substrate. Okay. So this Korai cycle, which was given by Korai and Korai in 1947, they received Nobel Prize. 80% of lactic acid which is formed. is reused, is reused. And how? Now I will show you. Okay. Here we write glycogen. Okay. Glucose. Glucose is converted into glycogen. Then pyruvate. We are just running opposite to glycolysis. Pyruvate. Okay. Pyruvate forms two molecules of lactic acid. 20% of lactic acid is lost in the form of CO2 and water. Okay. This is the liver cell and this is the blood cell, muscle cell, sorry. The glycogen enters the muscle cell, okay, forming glucose. Glucose is converted into pyruvate, using 2 ATP. Pyruvate is converted into lactic acid. This lactic acid moves to liver cell. Out of this, 20% is lost, 80% is, this lactic acid is converted into pyruvate. Pyruvate, which is converted into glucose, which is converted into glycogen. This glucose again enters the blood to supply glucose, the amount of glucose again in the muscle cell. So the fact is the muscular cell during external exercise, the muscular cells, the glucose is converted into lactic acid more because of less amount of oxygen depth. Okay. So this oxygen depth causes more and more amount of lactic acid formation. Because of more and more amount of lactic acid formation, starts to get accumulated, and this lactic acid try to decrease the pH of the blood. Now this lactic acid is converted into lactate, okay, and is transferred to liver cell to form pyruvate, which is converted to glucose, which is converted into glycogen. So this is the Cori cycle, okay. Around 80% of the lactic acid is converted into glycogen with the help of Cori cycle. Okay, understood? Now, muscular fatigue. It is the inability to contract muscle as accumulation of lactic acid desensitizes the muscle fibers. Because of greater or increasing amount of lactic acid deposition, okay, the muscles, they lost their ability to contract because of loss of the ability to release calcium ions from sarcoplasmic reticulum. Okay, so the desensitization of the muscle fibers because of the lactic acid deposition is basically muscular fatigue. Okay, Oxygen depth. What is oxygen depth? Oxygen depth can be understood during strenuous exercise. The muscles does not get sufficient oxygen. Okay. Sufficient oxygen to meet its energy requirement. Understood? The contraction of muscles under this anaerobic condition and the accumulation of lactic acid produced by anaerobic glycolysis. Okay? Accumulation of lactic acid produced by anaerobic glycolysis. Okay. During recovery, the oxygen consumption of muscle exceeds the extra oxygen consumed during recovery is called oxygen depth. So the reversal of all this phenomenon, whatever happened, okay. So during recovery, the oxygen consumption of muscle exceeds, increases, and this extra amount of oxygen consumed to convert lactic acid into CO2 and water, okay, is called O2 depth. Understood? The muscular groups. The different muscle groups we are going to talk about. For example, when we talk about the antagonistic muscles, okay? So a muscle these are the tendons. Okay. The origin, the end of the origin is fixed and movable end is insertion. Okay. There are around 639 muscles in human body, out of which 317 are paired, 317 muscles are paired, and rest are unpaired. Okay. So there is a category of muscles, synergistic muscles. Synergistic muscles, a muscle along with another muscle contracts to produce movement in same direction. Okay, you can say two muscles contract to produce movement in same direction. Then comes antagonistic muscles. Okay, these muscles are different sets of muscles which produce different movements. They are responsible for different movement. Okay, like when we talk about antagonistic muscles, examples are flexors and extensors. Flexors contract to bend one part over other, like biceps, and extensors, they contract to extend or open the bended part of the body, like for example triceps. So these are antagonistic muscles. Then adductor and abductor. Adductor move a body part towards the body axis midline. Example latissimus dorsi. Abductor moves a body part away from the body midline. Deltoideus. Levators and depressors. There are many antagonistic muscles. Okay, like masseter, it raises the lower jaw. Depressors, mandibularis, it depresses the mandible. Masseter, mandibularis. Okay, pronator, supinator, we have already explained these different types of movements when we were studying about the bones. Okay, so these are the muscle groups, synergistic or antagonistic. Okay, now we are going to discuss about, disorders. In disorders, the very first is muscular dystrophy. Although it is a genetic disorder, so it is a genetic disorder. Okay, dystrophin protein is not formed. Dystrophin protein is not formed. Dystrophin protein is present muscles between muscle fibers and necessary for coordinated contraction of different muscle fibers in a muscular bundle. Between for proper movement, for coordinated movement. Okay, it is a X-linked disease. X-linked disease. Okay, Duchenne's type. It is fatal by the age of 30 years. And Bakery's type or Baker's muscular dystrophy. It is milder. Okay, so this is the muscular dystrophy, which is the X-linked disease. Okay, it is of two types, Duchenne's and Baker's. Then comes Myasthenia gravis. It is an autoimmune disease. Okay, the antibodies are formed. Okay, and these antibodies, they are binded to acetylcholine receptors. Acetylcholine receptors. Okay, and the acetylcholine cannot bind to its receptors, which leads to muscle weakening and atrophy. Muscular weakening and atrophy. Okay, poliomyelitis. It is caused by RNA virus. The mode of infection is fecal-oral route. Fecal-oral route. Okay. The virus multiply in intestine and move to spinal cord. Okay. It destroys the ventral horns of spinal cord. Destroy the ventral horns of spinal cord. Where motor nerves are situated. When destroying the motor nerves. Hence, motor neurons cannot send message to the muscles, which leads to paralysis. This is about poliomyelitis. Okay. So, my dear students, we have already discussed different facts. Okay. There are two more three lines which I just want to share. Poliomyelitis, the disorders poliomyelitis, myasthenia gravis, and muscular dystrophy already we have discussed. Okay. Strongest muscle, masseter. Longest muscle, sartorius. Longest muscle group, quadriceps femoris. Largest muscle, gluteus maximus. Smallest muscle, stapedius. Okay. The strongest muscle is masseter, longest muscle is sartorius, the longest muscle group is quadriceps femoris, the largest muscle is gluteus maximus, and smallest muscle is stapedius. Okay, so these are the different muscles. Okay, I mean a good question for MCQ especially. Okay, now last we forgot to discuss about the hyoid bone. Okay, as the description is not given in NCRT, it is also known as tongue bone. So I have noted down tongue bone. Okay, it is located between lower jaw and larynx. Located between lower jaw and larynx. Okay, it does not articulate with any other bone. Does not articulate with any other bone. It provides insertion to some of the tongue muscles. Only provides insertion to some of the tongue muscles. Okay. The ear ossicles, as you know about Malleus incus and stapes, okay, they are present in middle ear. Hypomandibular, it is the fourth arch. The columella of hypomandibular forms stapes. Stapes is the smallest bone. Okay. Malleus is the modification of articulate bone. Articular bone. And incus is the modification of quadrate bone. That's it. Okay. So these are the some important facts. So my dear students, we have completed the locomotion and movement chapter. We have studied about the muscular physiology, the bone physiology, the anatomy of bones. Okay, everything we have discussed. So we will be meeting in the next term. We will start the other chapter of human physiology. Till then, bye bye. Take care. Stay safe. Stay healthy.